Hello and Tashdile to all the class 12 students. So in this video on chapter number 2 sexual reproduction in flowering plants, we are going to discuss about three small topics, and they are outbreeding devices, pollen-pistil interaction, and artificial hybridization. So first let us try to understand what do we mean by outbreeding devices. Now if you remember, in our previous video we have discussed about three types of pollination: autogamy, geitonogamy, and xenogamy. And if you remember their definitions, we know that in autogamy and geitonogamy, the pollen of a plant lands on the stigma of the same plant. Therefore, they do not bring about genetic variation. Right. Now, if there is continuous autogamy or geitonogamy in a plant population, it can lead to something called inbreeding depression. Right. So, inbreeding depression is not a good thing. So, we will see what do we mean by inbreeding depression. Inbreeding means the breeding of related individuals. Right. Inbreeding means breeding of related individuals. And if there is continuous inbreeding among related individuals, it will lead to something called inbreeding depression, in which the individuals they lose genetic fitness or genetic vigor. We can observe a reduced biological fitness among individuals who carry out inbreeding for a prolonged period of time. Right? So this is not good. It can lead to inbreeding depression. So continuous autogamy and geitonogamy leads to inbreeding depression, which is not a good thing. So in the plant, in flowering plants, in order to discourage self-pollination, which leads to inbreeding depression, right? Plants they have developed many outbreeding devices in order to discourage self-pollination and to encourage cross-pollination. Flowering plants they have developed many outbreeding devices. Cross-pollination means xenogamy. Just remember that. Okay, when the pollen of a different plant lands on the stigma of a different plant, that is called as cross-pollination, right? So it brings about genetic variation, and a genetic variation is good for the population. It avoids inbreeding depression. And the methods to encourage cross-pollination, they are called as outbreeding devices. Now let us see what are the outbreeding devices that plants have developed. So the first outbreeding device is asynchronous pollen release and stigma receptivity. Asynchronous pollen release and stigma receptivity means the timing of the pollen release and the timing of the stigma receptivity, they are not simultaneous. Right? Pollen release chayagi thuzu ta ani stigma receptive chayagi thuzu jo marre. Di manda chadu or yamena pollen nyela release chigre yamena stigma receptive nyela chagre. Di chidu zambala khar chigre la bana self pollination yoai chances ped ma nyor tangor. So that is the first one. And the second outbreeding device is the position of anther and stigma. They are different. If the anther and the stigma are located at different positions within the flower, it will discourage self-pollination. The third one is self-incompatibility, which is nothing but a genetic mechanism. In this mechanism, the pollen grain of the same plant cannot fertilize the egg of the same plant, right? The pollen grain, it is incompatible to germinate on the stigma of the same plant. So that is called as self-incompatibility. Kisi pollen grain meto chikaji ki pollen grain meto thakarang ka stigma gala chawa yin baina ani pollen grain di germinate cha tu mare pollen germinate ma cha yin baina ani fertilize che tu mar. So that is called as self-incompatibility. And the fourth one is diocy. Diocy is nothing but production of male and female flowers on different plants. Right? So diocy lawe na ngazu khala wore. Tishin chigi medo pod sugre, tishin shemba chigi medo moj sugre, and di nalo self pollination yoy ki chance kora yomarwa. So these are the 4 outbreeding devices. You should note down these 4 outbreeding devices and you have to know the details. Okay. Now the second topic of this particular video is pollen-pistil interaction. Right. Now we have already discussed about pollination, in which we have seen the transfer of pollen grain from the anther to the stigma of the pistil. Right. Now the pollen grain, after landing on the stigma, it undergoes certain changes, and these changes are brought about by pollen and pistil interaction. The pollen and pistil interacts. And how do they interact? The pistil, or the female reproductive part, it has the ability to recognize the compatible pollen. Right now, medo chigi ro penchashana rose medo gi pistil imbachasha di gangla ani medo shimba khaji sada bahar medo roji imbachasha odi gaji pollen dela tsawa imbaina ro species loga ro chik rose re chik sada bahar re dinde indu sambala. What will happen? The pollen, it won't germinate, right? The pistil knows that it is the wrong pollen, so the pollen is incompatible and nothing will happen. The pistil, it knows the right pollen and wrong pollen. So the pistil has the ability to recognize the compatible pollen. And it happens through communication between pollen grain and the pistil, and it happens through chemical messages. So phazu chemically phazu khonju phazu datang ni ani khonju ngoshingire, right? Correct pollen thadu mindu hagur. If the correct pollen lands on the stigma, right, post pollination events occur, and those post pollination events are called as pollen pistil interaction. We will see what happens, right? After the pollen lands, after the correct pollen lands on the stigma of the flower, the pollen, it germinates. Now pollen germination is very important, right? Pollen germinates to produce pollen tubes. So penetration they remove this here. You can look at this particular video, this picture, right? Here you can see the picture of a pollen grain, and when a correct pollen grain lands on a stigma, the pollen germinates through germ pore. Germ pore lana hakuro, where there is no sporopollenin. Exine la sporopollenin mesa ki area de langa khal lagyo germ pore lagyo. From a germ pore, the pollen, it will extend out a tubular structure called as pollen tube. Pollen tube la matangre. Ani pollen tube nanglola ani vegetative cell, the generative cell ni de matur. Ususo. And inside the pollen tube, the generative cell will divide into two male gametes, right? Vegetative cell whereas the generative cell, it will divide into two male gametes. I hope it is clear. This is called as pollen germination, and it happens only when the correct pollen lands on the stigma. Okay, so after it reaches the pollen tube, it sends the pollen tube towards the ovule. You can clearly see this pink structure over here. It is the pollen tube, right? So the pollen tube, it carries the male gametes towards the ovule, and when it reaches the ovule, it enters, the pollen tube enters one of the synergids. There are three egg apparatus, three cells in the egg apparatus, two synergids and one egg cell. Now out of those two synergids, one of the synergids will be entered by a pollen tube. Pollen tube khajire the synergids ni ne chignal ya zur, and it is held by filiform apparatus. Filiform apparatus ki rogo chigro and pollen tube dia nanglo ya zur. So the pollen tube enters one of the synergids present at the micropilar region of the ovule. So I hope it is clear. So the diagram, these diagrams are important. You have to practice drawing this diagram. So it shows pollen pistil interaction. When the correct pollen lands, it germinates, it sends out the pollen tube, and the pollen tube, it carries the two male gametes and the vegetative cell, right? And when it reaches the ovule at the micropilar region, it enters one of the synergids, right? So this is called as pollen pistil interaction. So you have to know the diagram, and you have to know the details of the pollen pistil interaction. Now in practicals also we will do the same thing. We will collect pollens of pea plants, chickpea, crotalaria, balsam, or winca. Pollen grain le, mature pollen grain le, and we put them in a drop of 10% sugar solution. 10% sugar solution now pollen grain is lura, and you have to wait for about 15 to 30 minutes. And after 15 to 30 minutes, if you observe them under microscope, you will see that the pollen grains, they have germinated. You can see the pollen tubes over here, right? So this is the experiment that we have to do in the laboratory later on, right? Successful young my own lading as the chena marwa. Okay, now the third topic is artificial hybridization, right? So artificial hybridization means, right, it is a method which is involved in crop improvement program kisi tyondo gi rig yar ke tangu yo bi ba na ta day gi rice plant. If you want to improve the rice plant, if you want to improve maize plant, right? So in crop improvement programs, artificial hybridization is used. So it is like an application of whatever we have learned in our previous videos, right? Manjogi male kajigi reproductive part ko la jangre, female reproductive part ko la jangre, pollination ko la jangre, raw pollination contest chungu me ko la jangre, any pollen-pistil interaction ko la jangre. Now, how will you apply these knowledge for crop improvement? Crop improvement like contest peju chigi me. So through artificial hybridization, if you do artificial hybridization, you can improve the crop varieties. In crop improvement program, in artificial hybridization, what we need to do is we need to make sure that only the desired pollen grains are used for pollination. Only the desired pollen grains are used for pollination, and you have to protect the stigma from contamination with undesired pollen grains. So there are two methods. There are two steps. Those two steps or those two methods are emasculation and bagging. Simply if you have got two varieties in a plant, zishin richig nalola medo manda ni yorda zishin chigi medo purple produce chigore, zishin chigi medo cow produce chigore. Now if you want to see what happens if we mix these two, zishin dingi ni mix cheni ngazula ni medo color khandi doji chungsare samgi kisi doba tado yobi imbai na. If you want to observe what happens if we cross these two plants, then you have to do something called artificial hybridization. In this plant, the flowers, they are bisexual. You can see they have both male and female reproductive parts. Now during artificial hybridization, first you need to decide which one you want to keep it as a male parent and which one you want to keep it as a female parent. Now after deciding that you want to keep this plant as the female parent and this plant as the male parent, first you need to remove all the anthers from the female parent. Anther yo digit sama ko or kachin ko or na anther mago imba na any self pollination chunga gi chances yoro. If you do not remove the anther, there is a chance that self pollination can happen. So in order to prevent that, first you remove all the anthers from the female parent. And after doing that, right, and the process is called as emasculation. The removal of anthers is called as emasculation. You can remove all the anthers. After removing all the anthers in the early stage, then what you need to do is you need to bag this particular flower. This is called as bagging, and bagging is done by using paper bags, butter paper. Why are we putting a paper bag on this particular flower? We are preventing any undesirable pollen grains from landing on the stigma. Stigma gala pollen grain shemba lands chimachu echu dungazu khachu wore digala butter paper yo. And when the stigma becomes receptive, when the time is correct, when the stigma becomes receptive, what we can do is we can collect the pollen grain from the male parent and then put it over here. This is called as dusting. And then you can bag it again. After bagging, the khachigre methodi swen le gyudure ro swen dinani khachigre kira plant khande dos ke do tayar and plant dige ki medo color khar do tayar. So this is called as artificial hybridization. Another very important topic. Right, only two methods you have to remember: emasculation, which is nothing but the removal of the anthers of the female parent, and then bagging, in which the female parent is bagged within a butter paper, right? And afterwards you have to take the pollen grain from the desired parent, male parent, and put it on the stigma and see what happens, right? You have got a hybrid of these two. So therefore it is called as artificial hybridization. Right, and it is used in crop improvement program. Okay, so in the next video we are going to discuss about double fertilization. So please read about double fertilization before you watch the next video. Thank you.